Start the broadcast, and it's the sounds of the opening cans and the flushing toilets.
Is that the sound it makes when you open the when you go live on a show? It's
like
beer opening. Oh, I bet it would have the same.
That's right. Kind of
a good sound effect.
Okay, we're gonna start this thing in 3, 2. This is TWIS. This Week in Science, episode number 622, recorded on Wednesday, June 7th, 2017. Planting a little seed. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we are going to fill your heads with some Einstein, old rock, and mucus magic. But first...
Disclaimer, disclaimer, disclaimer. There is an enemy of reason that seeks the destruction of us all. Stalks the most casual of conversations, lurks in the shadows of common misconceptions, gathers round the crowd to influence consensus, permeates the peripheral of popular perception. It is present in the lowest acts of inhumanity and at home in the highest, most hallowed halls of government. And this enemy, invisible to most, has a name. Ignorance. It's not a crime to be ignorant, nor is it a decent defense, but it is understandable why it is such a dominant force in the world and how powerless we may sometimes feel to defeat it. It seems an impossible task when each of us is ignorant of something. As if the alternative must be for everyone to be informed on every subject, to know everything in full expert detail so that we may be sure that reason is thriving in every instance. That we understand correctly the things we casually speak about. But the task is far less daunting than it may at first seem. We don't need each of us to know everything. We only need to learn how much knowledge is required to know the things we do not. By recognizing our own ignorance of a subject, we can be enlightened by those who are not. So, in an effort to become reasonably enlightened despite our own ignorance, we offer you This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
Ha! Good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back yet again to bring you a whole bunch of science news and an interview. It's going to be a very fun show today. What did I bring? I brought stories about general relativity and space voids. You know, as one does. What you do. We also have an interview with Adrienne Godschalx about defensive plants. Plants on the defensive. I mean, seriously, why are they so defensive anyway? I don't really understand. Anyway, we'll ask her these questions. Justin, what do you have for us?
I've got some early rock paintings. I've got, the oldest modern human fossil found ever. And I'm probably not going to do this one tonight. I'm going to tease it out, but this is going to be an ongoing thing. The CAR T cell therapy is doing incredible things right now. Oh, and they found the monkey cat.
Monkey cat! Oh my goodness. This is one for the record books going back. For those of you who have been watching TWIS or listening to TWIS for basically ever, you'll know what we're talking about. Blair, what is in the animal corner?
Oh, I brought some delicious digestive mucus that you were talking about before, and a little information on how we can help out with climate change.
Nice. We'd like to do that. I'd like to do that.
Yeah.
We do. All right. And let's see, I'd like to mention also this weekend, as Justin alluded to, is the Young Innovators Fair in Philadelphia, Pennsylvania. We will be at the Greater Philadelphia Expo Center Saturday and Sunday for the Young Innovators Fair. So if you are in the area, please come say hi. Catch the show. Maybe tell us about your science. If you're a scientist, we'll get you on. We have 2 days to fill with science, which is going to be very, very fun and exciting for all of us. And we will be trying to broadcast live on YouTube and Facebook. We don't know how it's going to work out yet, so we may just be recording it to tape for release later. But I hope you keep tuned to the social medias if you are not in the Philadelphia area, if you would like to check us out this weekend. We're going to see what we can do. We're going to do what we can, as one does. All right. It's time for our new segment in the show. What has science done for me lately? I'd like to read this email, for this Facebook message from Minion Jeremiah Foss. I am an avid outdoors person and live in eastern Kentucky. Thanks to the biologists in Kentucky, in my life, I have seen the return of the white-tailed deer to Kentucky, along with turkey, otter, beaver, and elk. Black bear are working their way back, and hopefully grouse and quail will make a comeback soon. Currently, they are battling diligently to stop white nose syndrome from destroying our bats and to stop the emerald ash borer from destroying our trees. I am sure there is much more that I am forgetting. Thank you for your message, Jeremiah. It is always, those are so important. It's nice to be reminded of the way that biology and ecology and the application of those sciences are helping to re-diversify areas of our country and areas of our world that have maybe lost those species. So thank you so much for that. And anyone...
And in that too, there's probably a lot more plant diversity or some changes going on in your forests and everything else. I mean, it's so directly tied. The story of Yellowstone is another great example of this, where the plant life in the forests started changing dramatically after reintroduction of species, starting with the wolf, but that has a catalyst for a lot of things that came after.
Yeah, many catalysts and many connections. For anyone else who is interested in sending a message and being a part of our segment, I'd like your messages. I like your emails. I like, send me a Facebook message, send me an email. My email is kirsten@thisweekinscience.com. You can message the This Week in Science page to tell us what you think science has done for you lately. I want to have at least one for every episode moving forward. I want a year of these, people. Let's keep reminding ourselves how important science is to our daily lives. Be a part of that. Join in. Come on, tell me your stories. And now it's time for our interview. You've probably noticed, if you're watching the video feed, that there's a wonderful young woman sitting down next to Blair, and she's just been sitting there quietly, waiting, biding her time. And you're like, who is this person?
I didn't know if I was allowed to talk.
Well, we'd love to introduce Adrienne Godschalx to the show. She moved to Portland in high school and studied biology at Western Oregon University. She's currently a PhD candidate in biology, getting ready to defend her dissertation work in plant defenses and biology. And she is a National Science Foundation Graduate Research Fellow in the Ballhorn Lab at Portland State University. Adrienne, welcome to the show.
Thank you so much. Thanks for inviting me, Kiki. This is such an honor.
You're welcome. I mean, we met about a little over a year ago, almost a year and a half ago. And I was really, really taken by the way you described your research when we sat down over a beer. And I was just like, oh my goodness, this woman knows how to talk about science. I need to get her on the show at some point. And then you put out a message saying, I'm getting ready for defending my thesis. And I was like, oh, let's practice.
Yes. Exactly.
So this is a TWIS style dissertation grilling.
Yay!
Bring it on.
Adrienne, just to get started, as a biologist, why did you end up studying plants instead of animals?
Ooh. Yes. Well, plants are a fascinating thing. Growing up in Moab, Utah, we didn't have many green things around us. And so when I moved out to Oregon, it was a very different world to see moss that could grow on sidewalks, on roofs, on tree branches. It blew my mind a bit. But the real story for why I'm now a scientist studying plants has to do with, in high school, coming from a small town, I wanted an excuse to get outside. And so I volunteered at a program called our Multnomah Education Service District Outdoor School, where I was a student leader and I was teaching sixth graders in the forest. Figured I'd babysat before, so I figured I could handle working with kids. But instead, I accidentally fell in love deeply with education and teaching about the river and about the trees that hold the soil on the riverbank. And from there, in college, I recognized the same lessons and just continued studying. And now I still do. I get paid to study nature, and it's incredible.
Do you think of yourself, I mean, you're in a biology program, but do you think of yourself more as a biologist or an ecologist?
I call myself technically a chemical ecologist. So a little bit of both. And I always joke with a few friends that the list of job names I could have on a business card, I want to get one with a scroll that would have statistician, biologist, ecologist, chemist, entomologist. I just continue accumulating terms that I identify with. But chemical ecology is neat because it is a term, and I brought some props for you, just so.
Great, we like props. It's
Okay,
always helpful.
plant. So this is what I spend most of my time at my desk thinking about. But when this plant is under attack, it can release a suite of compounds into the air, some of which, like this one, which is methyl jasmonate or jasmonic acid, a plant hormone, that when other leaves in the surrounding area get that hormone, those leaves know that there could be an attacker in the area, and they ramp up their defense level. And so the chemical ecology in that is that the shape of this molecule and the quantity that it's produced in, the ratio of various chemicals to one another can influence whether or not a plant gets pollinated. It can influence whether or not that plant is the most delicious plant on the block, and therefore the one that all bugs want to attack. And
so this molecule, it's released into the air, and it's kind of like, is this plant communication? Is this the plant telling the other plants, get ready, something's wrong?
Exactly. Yes, and it's not just one chemical, it's a suite of many of them. And all of them and in different ratios, they can mean different things. There are a few that we know for sure are plant hormones that have very specific functions that interact with one another, the same way that our hormones work within our bodies as well.
That's so fascinating. So in one sense, it's, I mean, another, you mentioned also whether this will determine whether or not a plant can get pollinated. So this is also interesting because you have insects and animals that can be pollinators, but they also can be attackers.
Exactly. Yes. So I spend most of my time thinking about how plants and bugs communicate. And so for my postdoc project too, I'm leaving the plant defense world, but not necessarily, because for my postdoc, I'm studying a plant pollinator system, but it's still chemical ecology because I'll be studying the quote unquote, we call all kinds of compounds that plants make that are not sugars, the basic photosynthesis product. We call all of those products secondary metabolites, anything beyond their first metabolism or making sugar. So I'll continue studying those secondary metabolites, but as the scent of certain flowers. So Kiki, when you think about the smells that are connected to plants, what are some strong smells that you can think of?
I love the smell of jasmine.
Yes.
I'm walking past and there's a jasmine bush. It's just.
So that's actually this. So jasmonic acid was discovered in jasmine, which is incredible.
So that smell is also, this beautiful smell from the jasmine flower is also, can be used defensively.
Yes.
Not just as an attractant.
Exactly.
That's fascinating.
And have antimicrobial properties. It can be directly toxic to many things. Yeah, volatile organic compounds, as we consider the tiny particles that do all the smell work, are they can have a vast range. And the funny thing about them too is that if you take one of these bonds and twist it, it's going to have a drastically different smell. So some compounds, the cis form is the active form versus the trans when the bond is arranged in a certain way. And so it can be very, very specific, but it can also be very, very broad, where the ratio of one compound to 20 other compounds might be the most important thing for a bee to recognize its host, or for an ant to be attracted to certain leaves compared to others.
Yeah.
The insect world around plants is like walking through Times Square. There's signs everywhere. There's flashing lights. There's things telling them to go this way, go that way, buy a ticket to the show. There's like a lot of stuff going on, not just looking around and seeing if a plant looks pretty and maybe that's where I'll.
Yes, it's like if you could advertise via taste buds. Like if we would walk around with our tongues out and receiving those advertisements.
I like you. I want to eat you. Oh, I like you. I want to lick you. just lick you.
It's not a good habit.
No. I don't know.
And I'm apparently the type of insect that does not like Cinnabon.
That's
okay.
True.
That's fair.
So
The smell that they have wafting out of there is totally noxious to me, but I know it draws a crowd of other human insects.
Which is a good point, actually, Justin. So if you think about it, we make a lot of our decisions about what we eat or what we don't eat based on what something tastes like, right? Mm-hmm. So the whole point of trying to understand plant defenses is because everything else out there makes decisions about what is delicious and what's not delicious, which can then result in major trophic food web changes, but is based on that smaller chemistry.
So in thinking about just the plant lifestyle, it really sounds as though what you're saying is that plants have evolved this very complex communication system to communicate among themselves and also with other organisms in the world around them. And their communication is something that can move through space in a way that they can't.
Yes, exactly.
All right. So what are some of these adaptations that what kind of defensive adaptations have plants come up with over the years?
Man, there's some good ones. So some extreme cases, plants can defend themselves in more than just these direct compounds. There are many plants that if you go into the forest, you wouldn't want to eat because they can be incredibly toxic. Some plants can make cyanide, like lima bean, which I study, or...
I knew I didn't like lima beans for a reason.
Now you have a reason.
Got it.
They're trying to kill you.
They are. Go ahead. Sorry.
And I mean, but most of our food tries to kill us too. Everything that tastes delicious is because that plant has been trying to not be eaten. Pretty cool. But plants can do things besides just make toxic compounds. They can also use, like I was talking about the insects, they can use those to extend their distance. Instead of being stuck in one place, they can bring in ants to that plant that can patrol the plant. And if you've ever seen ants at a picnic, they can be fairly aggressive. And when they find some kind of sugar resource, I find that ants tend to protect that resource. And so when a plant is the one producing a nectar, which is the case, plants can make, many plants can make this extra floral nectar, or a nectar that is used just for this purpose, to attract and draw ants and have them present on the plant, so that it wouldn't be very fun for any other organism to spend any time there.
I think one of my favorites was, isn't it catnip?
Oh,
that creates like the scent of like the sex smell of an aphid.
Ooh, possibly, yes. There's all kinds of stuff like that.
And they do it in the aphid off season when they aren't getting so it draws in predators for aphids to go and like look for them. Yeah. So just like you were saying, Justin, about the wolves in Yellowstone, predators can drastically change the community ecology. And so plants signaling to the predator, often plants can produce, just like you're saying, those hormones that will bring them in. And there are parasitoid wasps that
lay their eggs inside of, or they lay their young, their larvae, inside of the insect that's consuming a plant, which if you go to any rose bush, you can almost always find one of the aphids somewhere on that plant is probably puffed up like a Rice Krispie and a little bit golden brown. I posted a series of photos on Instagram and Twitter about this one aphid parasitoid that I found that we kept, and we put it in the lab, and this giant relative to the aphid, but it's like still miniature, like the size of a fruit fly, wasp came out of it. That was the young of a predator that had consumed that aphid.
And so is the rose doing something to actively like bring the wasps in to look for these guys, or?
We do know for sure that the parasitoid wasps are using the plant scent compounds to find their hosts.
To find their hosts. Oh, nice.
However, it's a messy world out there. So if you take one rosebush, one aphid, and one parasitoid in a lab, that works beautifully. But when you have aphids on a plant in nature that also is being attacked by a caterpillar and this other kind of larvae, there's a lot of different signals in the air. And an aphid sends attacking a plant compared to a caterpillar consuming a plant will send out different smells from that plant. So partly because they're regulated by different plant like chemical pathways. And so when one pathway is turned up, it actually crosstalks and turns off the other communication pathway. So the result is that the smell is not a perfect sum of both aphid-induced and caterpillar-induced. It's somewhere in the middle. And so the predators that can respond to that then vary, and then you have your community ecology determined by the top-down pressure varying based on those plant compounds that are released.
That's so fascinating. So let's go from here into what the focus of your dissertation work is.
Awesome.
Yeah.
So most of that is very similar. I have been lucky to be able to study almost all of what we just talked about. I study direct toxic compounds. I study plants using these, what we call indirect defenses, meaning bringing in predators to patrol and evict or kill off the other attackers, herbivore attackers on the plant. But not all plants are standalone in the soil. Many of them, especially plants in the bean family, Fabaceae, and I study lima bean, they are able to form a symbiotic relationship with rhizobia in the soil. And what rhizobia do, they're a bacteria that the plant roots send a chemical signal to the bacteria. The bacteria respond and send one back. And they basically give them a big tissue hug and create dip and dot like structures on the roots that then become nitrogen fixing factories. So what that means is our air is made up of about 80% nitrogen. But still, despite that, most plants struggle to be able to have enough nitrogen to produce the proteins and compete and make flowers and fruit and defensive compounds. So nitrogen is a pretty useful resource. And plants that have access to these nitrogen fixing microbes are able to use that resource in different ways. And so the exciting thing is that we found that plants with our lima bean plants that have nitrogen fixing bacteria produce greater amounts of toxic cyanide, but recruit fewer ants. And when I went to Costa Rica to test the native communities of parasitoid wasps, these same plants attract fewer of the wasps when they have the microbes. And so the big question that my dissertation work as a whole leads me to is whether these plants are actually limited, which let me explain. So any good relationship, even I'm sure among a team of 3 wonderful people that run This Week in Science, all relationships require energy and resources, and even the best ones. And that energy in the symbiotic relationship of bean plants looks like up to 30% of the plant's sugars can be directed towards the nodules, keeping that metabolism of the microbes, fixing nitrogen from the air and converting it into an organic form. And so it's a significant cost. And so one theory is that these plants are less able to attract predators because they're sending so much of their carbohydrate pool into nodules to maintain healthy rhizobia. My other theory is that it's more of a functional food web shift. Like we were talking about aphids and caterpillars, and there's so much going on above ground that if plants have access to that nitrogen and have the capacity to take it into their own hands, if you will, and make their own toxic compounds to protect their leaves, then spending extra sugar resources to bring in predators might not be as useful. Because even volatile organic compounds, they're not technically sugars the same way that the nectar would be to attract ants. But all of those compounds have often either 10 to 15 for mono or sesquiterpenes carbon molecules, which are derived from the carbohydrates made in photosynthesis. And so it might be more beneficial for the plant to be directly defended and rely less on those indirect. But that would depend on how the food web then would select for the most successful plants that could reproduce.
Isn't there also an effect on the surrounding plants when you have nitrogen fixers? So then they may be also either growing quicker or less quick. I'm not sure what it would be, but could they be drawing more predators away from that plant if they're suddenly more excited and growing quicker, but not having the nodules themselves to feed? Maybe they've got more sugars going.
You know, that's a very interesting question, Justin. Associational effects of whatever chemistry the legume itself is producing.
Yeah,
great question. I know for sure that the talking trees philosophy, or the paper that came out that was became big in pop culture, where one tree being attacked that then could communicate through the air to another tree. It's like, oh no, the trees can warn each other, crazy. Has later been translated as eavesdropping, where it is far more effective for I guess I have a real leaf, I don't have to have a hand leaf. Although the hand leaf is great. So if this leaf is attacked, and all of these leaves are now in danger of being attacked, it's far more effective to send out a compound into the air and be able to communicate to this entire cloud of leaves, especially if you're a tree with a lot, than sending a signal into the vein, down the stem, down to the stem, and into every single vein. Plus, a recent paper just came out this year showing that the vascular tissues don't actually connect to all parts of the plant. So if that were how plants did it, it wouldn't connect anyway. So to answer your question, Justin, the plants around in the area are able to eavesdrop from one plant's self-communication process.
Yeah, they can eavesdrop. But I was thinking also like the nitrogen in the soil. They got the nitrogen fixing microbes, right? Are the plants around it getting a benefit from this nitrogen fixing, even though they themselves aren't doing it? And if so, and they're not having to feed that resource, are they then allowed to produce more sugars and then, by course of that, be more attractive to the predators and pull away from the nitrogen fixer?
Huh.
That's what I would guess.
Good question.
Or would wonder about.
So the interesting thing, you're right, below ground is also a very complicated ecological world as well. It is not just root in soil with nodules. There are also suites of bacteria and fungi and all kinds of things, and nematodes living in the soil. So this entire above ground situation I've been describing has also been characterized below ground, but to a lesser degree. There's much fewer work because it's harder to see all of what's happening below ground. But to answer your question more directly, the benefit plants get around legumes, and the reason we use clover and soybean, all kinds of legumes as our cover crops for soil conservation and maintaining that ecosystem as much as possible, is not necessarily because the nodules are leaky. There is bringing nitrogen into the biological system is helpful, but part of that is through how would it get there? How would that nitrogen get into the soil? What do you think?
I have no idea.
Good question. So basically stuff dies. So as stuff dies, it gets into the soil and becomes part of that organic matter. The other thing that happens is the actual nitrogen fixation process produces hydrogen gas. And hydrogen gas contains energy that a different set of microbes can come in and break and use that energy, because nature's not about to waste that energy. Right. So the unique suite of microbes around the plants then in that soil community could influence how the neighboring plants interact below and above ground with all of their microbes and all of their
right, positively or negatively, depending on what that plant soil like sweet zone for a soil microbe combo is. Right.
This is the challenge of plant defense ecology now is the context dependency in, well, this works here and this works there. So understanding the mechanism for how certain factors like nitrogen fixing bacteria can influence a predator above ground, understanding whether that is actually a resource trade-off versus a plant allocation shift could be really useful, especially since many agricultural practices are excited and interested or possibly using biocontrol. This last weekend, I went to a wine bar and brought in my puffed up aphid and showed the winemaker, and she was very excited and talked about how they use biodynamic control, or how they, not biodynamic specifically, how they use this predator indirect defense by releasing parasitoid wasps to protect their vineyards.
Nice.
There's so much that we need to, you're right. I mean, this context dependency is going to affect so much of how we move forward in agriculture, in we put all this fertilizer into the soil to maintain the fertilizer, to maintain the soil and to have reduced soil degradation over time with farming practices. We've got these new practices with bio pest control, and there's so many things.
So many things.
So many that you would think this is a complex Earth system or something.
Weird.
Yeah. Tell me about Velcro.
Velcro. So the other cool thing is that plants don't just do everything with chemistry. They also have mechanical responses. So one of the exciting things is that sometimes plant defenses can show up in the same plants in force versus in others. So we found that our lima bean plants that have a lot of that toxic hydrogen cyanide also produce a higher density of these tiny little hairs that are hook-shaped that seem to act as Velcro that could be challenged. If you walk on a trail that has a tree down and all the branches are scraggly, it's going to be a lot harder to walk past to continue on your way than if that tree is standing up straight and right alongside the trail. Or even if you have to walk between the trees, it's easier than when all the branches are tangled in front of you. Mm-hmm. And so if you think about it, as a predator, mobility on a leaf could be important if the plant is relying on predators to come in and get rid of those herbivores. The other thing could be that herbivores interact differently with different defenses. So the cyanide that is increased in these same plants that have the tiny hook trichomes or hairs, they're very well defended against chewing herbivores by having that toxic defense. Because the way that cyanide works or cyanogenesis is the name of this defense, it works like a glow stick, where there's a compound inside most of the cell in that big vacuole that fills most of the cell. But there's an enzyme that plants make that's determined genetically that is in the surrounding apoplast. And so when the glow stick is broken, those two compounds mix and the enzyme cuts a glucose off of the hydrogen cyanide and releases that as a toxin. So for herbivores that do the mashing and they grind up the leaves effectively, it releases that and allows it to happen. But an aphid kind of sneaks its stylet in and goes between the layers and doesn't necessarily rupture as many of those molecules. So by having another defense co-vary and show up in the same plants that are highly cyanogenic, those plants could be more defended against multiple feeding guilds.
I think it's kind of like the movies where they do the macro lenses to take you down to the level of the insects. and you start thinking of the world that these plants really are living in. I mean, I'm living in this world and I'm like, oh, look, a bird, a tree. That's wonderful. But these plants are living in this much tinier scale. They're having to pay attention. You know, I'm like, oh, an ant or a flea, and you flick it off, right?
Yes.
But they're really concerned about those mechanical defenses, the chemical defenses, and how they can work at the large scale and also at the small scale.
It's an unending biological arms race.
Beautiful. That's beautiful, Blair. Yeah.
So you have a question in your blog, one of your blog entries that's here. It's, should plants protect their flowers?
Yes.
I think they should, because flowers are pretty, but
Well, flowers aren't fruit, right? Fruit is meant to be eaten, but flowers are meant to be pollinated. So I guess you don't want flowers to get eaten, right? You just want them rubbed up against if you're a plant. Isn't that right?
Yeah. Yeah, there has to be some rubbing for the sex to happen. You're right. And
you heard it here.
man, if that's my one tweetable quote, I'm winning.
that's right. When we put out the show, I'll have that as the quote. No.
It's actually now the title of the show, today's show.
So that's a complicated thing, right? So if the plant needs to bring in the right bugs, it has to filter out for the right bugs.
Mm-hmm.
But creating something that is attractive is going to attract most things. That paper was fascinating because we just assumed, sure, flowers lead to seeds, and natural selection acts on anything that helps the plant make seeds and survive and bring its genes to the next generation. So if flowers are demolished, then one would think that they would be less successful. But in the case for lima bean, when we tested the actual cyanogenic potential of the different ages of leaves and flowers, we found that the flowers weren't very toxic at all. And the highest amount of cyanogenic potential was found in the young leaf tissue. And those young leaves, so we're like, oh man, that doesn't actually line up with the optimal defense theory or the most fitness related pattern that we would expect to find. And so the interesting thing is when we actually removed the flowers and tested it, that there wasn't any fitness effect, that the plants made just as many seeds. So as long as we left some flowers, it didn't matter how many they had, which was fascinating.
So they just need some.
Correct.
They don't need all.
And sometimes there's a compensatory growth effect where the reduction can focus the energy into certain plants. So the interesting thing came when we did the same thing with the most toxic organ, when we took young leaf tissue away. And young leaf those are the potential of the future, the photosynthesis of days to come, when days are hopefully better for that plant.
And you told the plant, I don't care about tomorrow. What can you do for me right now?
So we removed different degrees, quantitative levels of leaf tissue, and found that the number of seeds that plant was able to make dropped with it dropped along with our leaf removal experiment.
Oh, wow.
And so it actually quantitatively affected fitness.
Wow.
Which is pretty neat. So it did show, in fact, that yes, plants are putting their money where the fitness is. Essentially, they're spending their cyanogenic potential where the fitness benefit can come from.
Right.
But it took us testing that to actually have that insight.
So the plants, the flowers can get eaten up to a certain extent, but the leaves really, they need to stay.
Right. They're, the currency of the future, and I suppose. The interesting thing too is that it's not consistent across plants, that other plants would have a different effect in theory. I found a lot of cool papers as I was working through that one. So if you're excited about flowers and different strategies of where a plant should put its most valuable poisons, it's a fascinating area.
Yeah. So do you think much about, the toxin like cyanide, people don't want to eat cyanide. So we don't eat lima bean leaves, right? We eat the lima beans. We don't eat the lima bean leaves. But there are other compounds that we do really like. And what is it about various compounds that, I mean, why are humans different from other animals or primates, I guess? What are we, why are we like, I like that bitter flavor. I'm going to eat that kale.
Well, do monkeys not? I don't know that.
I don't know.
I've never given a monkey kale before to find out.
Blair, have you given a monkey kale? You have?
Did I eat it?
Yeah.
Okay. So this taps into a question that I grappled with the first few years of grad school. I still kind of do, because it baffles me that these same compounds that I study show up on, like, there's a burrito place in Portland, Laughing Planet, that sometimes has a sign that talks about the value of isothiocyanates. I'm like, wait a minute, but that's the defense compound in those plants, or glucosinolates, that is in kale and allows them to be so healthy. So why is it this plant poison that is conferring our human health benefits? And you can find all kinds of research. I've ended up in an internet wormhole.
I'm sure.
But my gut reaction is that the reason those compounds are useful as a defense is because they interact with biological systems. They're not inert. They have some kind of reaction. And so there was an interesting work done where I think it was a German group that injected chloroplasts that put them into a cancer cell. And of course, the free radicals they generated just destroyed the cancer cells. And I'm not by any means any kind of cancer biologist or have much of that expertise, but I do know that alkaloids are able to interact with nervous system receptors. And so that's how they have that caffeine effect. But that's also why they're toxic and why the plant makes them to begin with. I think the biggest thing here, why we can eat many things that would be toxic to a caterpillar, is
we're not a caterpillar.
We're enormous.
Yeah.
Exactly.
We've got a liver and kidneys.
It's true.
Right.
And sometimes the caterpillars can specialize. They can choose to consume caterpillars, aphids. There are specialists on almost everything that can choose to consume the most poisonous plants because they either have figured out how to detoxify them or can store them and then allow them to succeed more. And like the monarch butterfly situation, where then they are not a delightful treat and the birds quickly learn to not consume monarchs. Long live the monarchs.
And the monarch mimics who don't actually taste like them but look like them so that they get that benefit of not getting eaten.
Exactly. And then they don't have to deal with creating the enzymatic cost of actually dealing with the toxins.
I love all this. So you are, when are you defending your dissertation?
Friday afternoon at 2 p.m.
Friday, you're almost there. Congratulations. And where do you go from here?
From here, I will do 2 more experiments on this system over the summer that I don't have to do for my PhD, but I'm so curious about what would happen. So I'm kind of just having some fun projects that will, in theory, stock the data fridge.
Mm-hmm.
And I love teaching. I see research as research and discovery and creating new questions as a vessel to teach and help share. Somewhere along the way, I figured out this very nerdy but beautiful affinity for the natural world that I can't not see. And I feel like I really need to share that. And in any way that I can help anyone else see the beauty around them, whether it's in the way that the Krebs cycle works or how plants can make certain compounds, or any part of how this beautiful natural world works, I want to share. I want to illuminate how the world is beautiful. So a more tangible answer for you is that I took a postdoc in Switzerland and will be heading over there in August, studying a beautiful flower that has an incredible deceptive pollination system. What they do is they smell like poop. and flies looking for poop to lay their eggs get trapped. They fly in, and this incredible system has a narrow part in this flower where the flies fly in and they buzz around the females, pollinate them. The males bloom and shower them in pollen. The flies buzz out 24 hours later and get tricked again. And so my big question is looking at the poop scent profiles and figuring out if they differ in Italy to the ones that are in France, to the ones that are in Croatia.
Oh.
And see if the fly species that's most attracted to those specific scents is actually the one that's pollinating them the most.
So do you think that's
Right.
maybe because there are different animals that are pooping in those areas, and so they might have different scents to relate to those
Oh
kind
yeah,
of
probably, which is
scent
probably really,
profiles?
yes, and possibly the food that those animals ate in order to create, and their microbial processes. So maybe it is another bottom-up effect of a
It's
microbe.
all connected, man. It's all
Now you can go around and say, yeah, my postdoc,
Poop scent.
poop scented flowers.
I do. My friends love it.
That's fantastic.
The emoji world has already set me up for success. I'm excited.
It has,
you're all set.
absolutely. Many poop emojis in your future.
Yeah,
So
great.
is this the scent compound? Is there one compound you're looking at, or do you already have molecules in mind, or are you doing a chemistry treasure hunt?
A little bit of both. So we have, as a community of chemical ecologists, we have like a set of characters that show up in many plants and do certain things that we're like are buzzwords. So we know jasmonic acid is related to chewing herbivore defenses, or certain compounds like linalool is a very flower pollinator system compound. But we're learning more and more about what each of those compounds can do, but also the broader scale of how everything else around them actually matter as well. So a little exploratory, but I'll keep my eye out for some of my old friends that I know well.
Nice. Very nice. And you'll be in Switzerland.
Yes,
some of the time. So are you speaking French, German, or Austrian?
I will be in the French region. There are 4 languages I've learned in Switzerland.
Oh, a fourth.
Yeah. It's a Romanche. So it's Italian, French,
Italian, that's what it is. Yes.
and Romanche. Yeah.
All right. You're
It's going to be great. I need to learn
fluent
French.
you're fluent in all of them, right?
Yes. I'm fluent in smiling and figuring it out.
That is a great skill. That's where you start. And finally, getting into social media and your communication efforts. You have an Instagram account, you have a Twitter account, and you have your website with its blog. Can you tell people where they can find you?
Yes. My Instagram is Godschalx, which is my first letter of my first name and my last name, Godschalx. And the my blog and website have come from actually working. Part of where I developed the way that I talk about what I talk about has come from working at a restaurant and bartending. And as I would make a mojito, I would notice that guests would get excited as I talked about, well, you know that these mint compounds or the alpha limonene in your lemon twist are actually the result of plants in a long coevolutionary battle with a bug that was trying to eat it. And so I've generated a lot of really fun conversations across the bar top, which is how I came up with the concept for a blog called Science Martini. My tagline is Plant Biology with a Twist.
Nice.
Well done.
Nice.
Thanks.
And Ed from Connecticut wants to know, what's the plant biologist drinking game?
Great question. Well, Ed, I challenge you to make a cocktail that is out of some sort of either wintergreen or jasmine flavored something. That's not as, it's a game for a bartender. I don't know if it's a game for everyone in the world. But wintergreen is the smell of that methyl salicylate, one of the plant hormones that is released typically in the aphid world. or in response to fungi that get inside of a leaf, which we also didn't talk anything about, but that's a whole other world. That you'll have to talk to my buddy who also is in the lab, Brett Younginger. So he studies them.
We like the fungi.
Yeah, I hear he's a real fun guy.
Blair cannot leave a pun. I can't leave the pun standing. Adrienne, thank you so much for joining us tonight. This has just been wonderful hearing about your work and hearing about where you're going with it. And good luck in Switzerland.
Thank you so much, Kiki. This was such an honor.
You're welcome. Everyone, once again, you can find her at sciencemartini.com or Godschalx. We will have links on our website, twis.org, if you don't want to remember these things. So if you're interested in, also her Instagram account is full of some fabulous pictures. I want you to know, I love following your Instagram.
Thank you. I appreciate
that. I like going through it. I'm like, oh, beautiful nature photos. Yeah, it's always
there's always time in the day for wonder, right?
Yeah, exactly. You do a good job with that. So I highly recommend the Instagram for everyone out there. All right, you guys. Yes, great guest. People in the chat room. Thank you so much, Adrienne.
Thank you.
You're welcome. We are going to take a quick break and then come back again for some science stories. This is This Week in Science. Stay tuned. Hey, everybody, this is This Week in Science. I hope you have been enjoying the show. We just had a wonderful interview, and we hope to bring you many, many more. I want to have lots of people talking about the science they do and helping to fill you with wonder. And if you want to hear lots more wonderful stuff, we will be at the Young Innovators Fair this weekend, Saturday and Sunday in Philadelphia, Greater Philadelphia Expo Center. Saturday and Sunday this week. Oh my goodness, the 10th and 11th. I'm leaving on a red eye with Justin tomorrow night. It's going to be amazing. I'm going to be so tired. We're going to be so tired all weekend, but it'll be great. But, you know, it is support from people like you who listen to the show that allow us to continue to bring great interviews and science news to you week after week, and to do fun things like heading out to events like this Young Innovators Fair. It is your support that allows us to produce this show, to have the equipment that we have, to really keep this ship running. And we thank you for that. And if you're interested in supporting us, you can head over to TWIS.org, and there are a couple of ways that you can get involved in helping us produce this show. One of the ways is to go to our Zazzle store. In the Zazzle store, we have all sorts of the link is in the main header bar at twis.org on our website. You click on it, it'll take you to our Zazzle store where we have all sorts of wonderful items with the TWIS logo, with art from Blair's Animal Corner, from her yearly calendars, and there are stickers and mouse pads and mugs and aprons and tote bags and hats and sweatshirts and t-shirts, all sorts of things. Go peruse, enjoy, and we hope that you go home, or that something is sent to your home from the Zazzle store, because part of the proceeds from all sales do go directly to supporting TWIS. Additionally, at twis.org, we have a donate button. So if you would rather just click on that donate button, you can do that, and it will take you to a PayPal page where you can use PayPal to send us the money. Yeah, it's very, very easy, pretty painless, and it helps us immensely. If you want to get involved in our Patreon community, you click on the Patreon link in the main header bar. That will take you to patreon.com/thisweekinscience. And you just click on the big orange Become a Patron button. It's a great big button. You can't miss it. And if you click on that, you choose your level of support, and it's on episode by episode basis. And you can set limits for the month, though, and you get little rewards for being a part of it. If you heard at the end of every show, I read off a bunch of names, and those are the people who are pledging $2 or more per episode. So if you pledge $5 an episode, you will get your name read as well. All sorts of neat things here on the Patreon community. We do occasionally share special content, little notes and messages with our Patreon community that don't go out to everybody else. So it's kind of like a little special place that you can be a part of. And while you're at it, you help produce TWIS. You really help us make this show possible. We thank you for your support. We could not do this without you.
It's hard. The methods and hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
And we're back with more This Week in Science. Kiki, what you got?
I have general relativity.
Mhm.
Yeah. I got general relativity, you guys.
We all have that, right?
We all have that. Well, Einstein came up with the idea, right? We can all rest easily now that we know general relativity. It's like, oh yeah, general relativity. And this is the standard idea of how the physics of the universe works and how energy and everything are related, mass and energy and all that kind of stuff. Well, there's still questions about how it really works and is it really right? Does it explain everything? Lots of questions about that. And so there's a very interesting study that was reported this last week, and a group out of, where are they from? A group used the Keck telescopes to look at a couple of stars that were orbiting a big massive black hole here in the Milky Way, and looking to see how that black hole affected the light around the stars, affected the gravitational movement of the stars. And, there's certain predictions that can be made based on general relativity. And if general relativity works, you'd expect one result. And if there's something wonky and there's something else at work there, other factors at play, then you would see a different result. And so this team that dug up this data and did this test around this supermassive black hole here in the Milky Way, they have about 25 years worth of data that gave them about 100 or so observations from these different stars. And they had all this data that was each time, because it was over like 25 years, the observations were performed a little bit differently. So at one point, it was just a simple observation. Then the telescopes were upgraded and there were adaptive optics added. And then there were spectral data obtained as opposed to just imaging. And so the spectral data, you can see actual Doppler shift because of the movement of the star. And so that gives orbital velocity information. And so all of this was put together. And these scientists were like, okay, we'll take all these different numbers and all these different measurements and put it all together. And we want to see how this all works. And basically, they confirmed general relativity. Nothing was weird. Everything was just right. So general relativity wins.
Again.
Again.
And again and again and again and
Yeah.
and again and again and again.
And again and again and again. Yeah. So the big thing about this is that it's neat that they've been able to take this old data, but the way that they were able to look at it was because this was digitized open data. These were data sets that other scientists, those who work at the Keck telescopes, made available to the public. And so these scientists were able to go, hey, I want to look at this. Oh, look, there's a database. There's this data, and we can data mine it. We can find the data that we need within this data set that's been collected for 25 years.
So rad.
Yeah, and whereas once upon a time, it was like, oh, we took one measurement and it was a picture, and then we took that picture and we put it in a filing cabinet somewhere, and that filing cabinet is in Antarctica, and nobody else can see this picture ever again.
Yeah.
So the way in which this study was done is a very exciting movement, and it's not just astronomy that's moving toward these open data sets. There's also climate change. A lot of physics is moving this way. The Large Hadron Collider at CERN, their data is big open data sets so that data mining can take place to be able to pick out information and be able to figure things out about the universe that maybe we wouldn't have been able to before because all that information was not stored really well or just shoved in a filing cabinet, and maybe it's all gone after that scientist retires, right? And then additionally about it, that it's neat to be able to test these ideas about gravity using these massive black holes in space and how they affect the movement of stars, because we really can't do those tests in labs here on the planet. It doesn't work the same way. So we have to look out away from ourselves to be able to and use high-powered telescopes to be able to really get high-resolution confirmation of ideas. Like general relativity. There we go. Wins again. University of Wisconsin-Madison researcher Amy Barger and an undergraduate student presented at the meeting of the American Astronomical Society this week on results of their new study looking at the Swiss cheese of the universe and the hole in that Swiss cheese in which we live. We live in a big Swiss cheese hole.
You're a Swiss cheese hole.
I know. So our universe, we know that it looks like a bunch of filaments, these images of what our universe looks like as we've simulated it. It looks kind of Swiss cheesy, or like a thick cobweb where you have filaments of matter, stars and dust and gas that tie everything together. And then in between those filaments, you have voids. And the voids don't have much in them. However, the Milky Way exists inside one of the largest voids in our universe.
We live in the Boondocks. Way, way out there in the fringes.
Yeah, we live way out. It's not just the fringes, but we live like in the desert. Like there's not much around us, we're out there, man. Way out there. And so what is it about being in a void that is important? And so what this new study really looks at is it really confirms, or more closely confirms, the fact that we are in fact indeed in a void. And as a matter of being in the void, we're not seeing the universe consistently every time we look at it, because sometimes we're looking at things that are in the filaments where there's a lot of matter, and other times we're looking at things that are more close to us within the void. And so the expansion of the universe, and we've talked about this before on the show, there is not always consistency in the measurement of the rate of expansion. And the reason behind this is that we live in a void. And so the void it changes the answer we get depending on what observational technique we use. So what this study basically said is that it brings all these measurements and kind of equilibrates them, so that because we're in the void, there are certain factors in the void where we're not seeing expansion as much. This, the speed of light or speed of objects moving away from us is not as rapid. In the filaments, we're seeing things move more rapidly to that degree. Yeah. So the Hubble constant is the main measurement that they're looking at. And so the researchers say that a direct comparison can be made between the cosmic determination of the Hubble constant and the local determination derived from observations of light from relatively nearby supernovae. So we have supernovae standard candles that we use. We look at the light that's traveling to us from those supernovae. We know how brightly they burn, right? And so we know how much light and how bright and how far away they are as a result of that. But that's more local. Yeah, it's more local versus, say, the cosmic microwave background radiation, which is this cosmic stuff, and it gives a different answer. And so there's this new analysis that shows that there are no current observational obstacles to the conclusion that the Milky Way resides in a very large void. And the presence of the void can resolve some of the discrepancies between techniques that we're using to clock how fast the universe is expanding. Knowing where you are makes a difference in how you look at everything around you.
Relatively speaking.
Relatively speaking.
That sentence can mean a lot of things. Knowing where you are can have an effect on how you see the things around you. Yes.
Mm-hmm.
Absolutely.
Absolutely. Well, it's in relativity, so I'm on a bus. Things are moving. No, those things are stationary. I'm moving.
Mm-hmm.
This understanding, knowing you're on that bus, is going to change the way that you perceive what's going on around. And so it will change the way it equilibrates, the way these calculations are made.
Mm-hmm.
So pretty fun stuff, huh?
Trippy,
man. We're in a void.
Yeah.
It's a big cosmic
Far out, dude.
void.
One void.
Yeah.
Oh, yeah.
Go ahead. I was just going to say that we could stay in our void and study poop flowers.
Okay. I was going to say that one void in our knowledge about the age of mankind, Homo sapiens, has been filled in. Age of modern humans just got a little bit older. Actually, it's exactly as old as it ever was. But now we have a better idea of just how ancient we really are. Fossil bones, stone tools dating to about 300,000 years have been discovered. And it's not where some might have expected to find these ancient humans. These were found in Morocco. Previously, the oldest Homo sapien fossils were found in Omo Kabish, site in Ethiopia. Those dated at 195,000 years ago. Until now, the working model was that all humans living today descended from a population that lived in East Africa around that 200,000 year-ish time. Quotey voice. We used to think that there was a cradle of mankind 200,000 years ago in East Africa. But our new data reveal that Homo sapiens spread across the entire African continent around 300,000 years ago. Long before the out of Africa dispersal of Homo sapiens, there was a dispersal within Africa, says paleoanthropologist Jean-Jacques Hoblin, who probably sounds nothing like that whatsoever. Though we have covered on this show before, there was a modern human integration, if you will, into a Siberian Neanderthal lineage that occurred from 100 to maybe even 230,000 years ago, as seen through the DNA of a Neanderthal fossil found in Siberia. So already we had clues that our origins may be a little bit older than the oldest fossil find, and perhaps much more far-flung. That and when finding fossils, there are places where fossils form easier, and areas where fossils formed are more accessible. East Africa is the sweet spot for both of those scenarios, so it's sort of the low-hanging fruit of paleoanthropology. The whole Rift Valley and also the area where the oldest fossil is found are areas in which you have in access to, sort of like if you were looking at the sides of the Grand Canyon, you can sort of go back through the years and the ages, and because it was sediment and flood zones, these are areas that would cover fossils well, and then also make them easier to find, being in an area that was accessible years and years later. So that said, this finding of a fossil 100,000 years earlier than the previous oldest find is like breaking an Olympic record, not by tenths of a second, but by 2/3 of the old time. This is a huge discovery. Wow. The Moroccan site Jebel Irhoud, I have no idea how to pronounce it, has been well known since the 1960s for its human fossils and artifacts. However, the interpretation of the 22 fossil remains of skulls, teeth, long bones of at least 5 individuals remained somewhat unclear. It had been estimated to be 160,000 years old, so not nearly as old as they're now finding. So to provide a precise chronology of these finds, researchers used the thermoluminescence dating method on heated flints found in the same deposits. These flints yielded the age of approximately 300,000 years. This is quotey voice of somebody else. Well-dated sites of this age are exceptionally rare in Africa, but we were fortunate that so many of the Jebel Irhoud flint artifacts had been heated in the past, says geochronology expert Daniel Richter, Max Planck Institute. He explains, this allowed us to apply thermoluminescence dating methods on the flint artifacts and establish consistent chronology for the new hominin fossils and the layers above them. In addition, the team was able to recalculate direct age of a mandible found in the 60s. This mandible had been is the one previously dated to 160,000 years and pushed it up to that 300,000 year range. Also, what's really interesting, the crania of modern humans living today are characterized by a combination of features. This is morphology stuff that distinguish our fossils from our ancestors and maybe some of the cousins that were running around. The morphology and age of the fossils from the Morocco site actually are corroborating an interpretation of a partial cranium find from Floresbad, South Africa, that that was thought to be an early representative of Homo sapiens, but there's been some it wasn't really confirmed that this was an early Homo sapien. They only have this partial cranium piece. That piece is estimated to be 260,000 years old. So it's not just the one pushing it back to 300,000. Now, because of morphology, they're connecting it to a previously dated site that's at 260,000 in South Africa. 2 outliers from East Africa now, much, much, much older than the previous oldest modern human Homo sapien ever discovered. So that's pretty awesome. They say the fossils were found deposits containing animal bones, showing evidence of hunting. Turns out our favorite meal back then in Morocco, gazelle. Everybody loves gazelle. It's good stuff. Most stone tools were made from high quality flint that were actually imported to the site. The flints themselves couldn't be found there. Also interesting, hand axes. A tool that was commonly found in ancient sites that aren't that ancient, were not present at all at the site. So we may have a little bit of a timeline for the origin of the hand axe amongst modern humans now.
And Ed from Connecticut is saying we need to shout out to UC Davis for this study as well. UC Davis was involved in this study.
Oh, that I didn't know. I wasn't in what I heard.
Yes, absolutely. I think that's the first that I had heard about it, was a UC Davis report.
Yeah. And other oldie stuff news. There's this is Professor David Pierce, director of the Rock Art Research Institute, University of Witwatersrand, Johannesburg. They managed to carbon date some cave paintings that they found. One in southeastern Botswana. They're at least 5,500 years old. And some in Lesotho, Eastern Cape Drakensberg, South Africa, that are as old as 3,000 years. So this has been something they haven't been able to do before, is date these paintings. We're, I guess, not talking about an artifact site that's that simply found, but they did carbon dating. They removed a millimeter of the actual paint. They took it, and they managed to date it to that ancient age. Some of the site's paintings continue to be made for more than 1,000 years. So 1,000 years, people are coming back to the same caves and adding paintings to them.
1,000 years of graffiti. Could you imagine if people kept putting gum on the gum wall in Seattle for 1,000 years? Not the same.
So you're saying graffiti. So this is Pierce quote. People return to the same rock shelters over long periods of time to make rock paintings very similar to those made centuries or millennia before. This finding has profound implications for understanding of hunter-gatherer religion in Southern Africa. And I take a little bit of umbrage to both graffiti and the religious aspect of this. This is only a personal observation, but while it's common for cave paintings to be interpreted this way, there seems to be very clearly an educational value to the artwork. There's tales of caution. Every once in a while, somebody's being gored by the horns. Watch out for that end. It can be dangerous. There's hunting methods, and it's a zoological record. Remember, not too long ago, we were talking about cave paintings in Europe, where they discovered that actually there was a bison that was a hybrid of two others that was actually being correctly portrayed in cave paintings, whereas it had looked like they'd kind of just been rough shot artwork where they were getting elements of the bison that are there currently wrong. So for 1,000-year site, we may be looking something that's much more akin to the first library or university, and stop thinking of it purely in religious context. We took lessons from animals early on. I mean, this is how we found sources of food and where to go for water and shelter and these sorts of things. So it's studying the life forms that were present for early man was the biggest, most probably important form of study that existed. And it's probably the first science that we're looking at.
And they're dating this back to 5,000 or 3,000 years, right? So these
one site is 3,000 years. One went back to 5,500 years.
Yeah. So the timing of this versus like Egyptian hieroglyphs, where that was also historical record, educational. This is, they're writing down with their hieroglyphs.
History and pornography all took place.
All of it.
Yeah.
Yeah. So this, and that's
satire
probably,
was there.
and it's around the same time period, right?
Ish. 5,500 years old is older.
Yeah.
But ancient Egypt's around 3,500, I think. Yeah.
3,000 years is kind of
But I think that a lot of early writings, and anytime there's a new methodology, often falls to religion. So I understand why
it's got to be religion.
Why that's a hypothesis.
I think it's a presumption. I think it's presumption.
It's sure, but it has to do with the fact that some of the very first books ever printed were of the Bible, right? So when that technology exists, I don't think it's a huge leap to go there. But I also think that just recognizing it as a written account, we don't know exactly what it means, but recognizing that it's definitely a recording that's not just for fun. Mm-hmm. I think that's important to know.
And there's books much older than the Bible, of course.
Mm-hmm.
Like much older.
Yes, but in terms of mass production, it was one of the very first.
Well, yeah, okay. For you're talking printing press.
So I'm saying that when a new a new methodology rises in recording and spreading information. It often is used for religious texts. That's pretty common.
I don't know. I would just totally disagree when it comes to the Greeks.
we call it religion.
Totally disagree
but
when it
it's
comes to the Greeks.
culture.
But, you know,
Yeah, it's
fire,
culture.
you know,
Absolutely.
also,
Whatever
it's a great equalizer and inquisitions and things like this. But moving on.
Yes, please. Let's keep going. We're gonna wrap this show up.
I'm only going to do this really quick. Chimeric antigen receptor T cell therapy. They did a test in humans. They were working with multiple melanoma, and they had a 94% clinical remission of multiple melanoma upon receiving myeloma upon receiving this new immunotherapy. So this is where they take your own cells out. They genetically reprogram them. One of the versions of this I've seen, they were actually using HIV, getting rid of the dangerous parts of the virus, reprogramming it to attack certain types of cancers, putting it back into the body in with hours or days, curing people. Thus far, this looks like the side effects were mild in previous versions of this, and for a different use, it was actually killing a small percentage of the patients who were mice, I think, at the time too. So, oh no, actually humans. They actually did a human trial, and it was humans that died from this. Other people have been cured. So, but this is CAR T cell therapy. We're going to be talking about this more in the future. So today, this is just sort of a teaser version. And Borneo, formerly Borneo, Kalimantan, they got footage of the monkey cat.
The monkey cat.
The monkey cat is real. Now they're still claiming this is more cat than monkey.
Likely. I saw the
At this
pictures.
point.
Definitely looks like a cat.
Well, it looks really monkey-ish to me.
It looked lanky and a little monkeyish.
It's
I think
got
it's just
a long
a cat with a tiny head.
A cat with a tiny head. It's got a kind of a bulbous butt that's sort of monkey-ish. And, you know, that tail looks like a monkey tail. But, yeah, it looks like they're calling it a bay cat. But this is something that was captured a decade ago, plus maybe, in a few frames of a motion detector camera with low resolution at night. And
we were still at UC Davis down in the basement at Freeborn Hall when the monkey cat images were first taken.
Yeah, and this is quote from the Honest to Pause site that this got sent to me from. Just wait until you see this sleek feline in action. He almost looks like a cross between a black panther and some sort of giant monkey. Right? That's monkey cat. That's finally discovered. And they actually have a video.
It doesn't even look like a cat to me. It looks like a weasel.
Weasel.
Yeah.
That's interesting.
Now you're gonna start a whole new drama of a monkey weasel.
Monkey weasel.
I'm very curious. I was looking at this because it did get sent to us from this website, but there's not really any cited material. Because I was trying to figure out what they think, genus or family or anything related
to that.
Well, they're
calling it bay cat in the version I saw.
Right.
I saw they're calling it bay cat.
What does that mean?
And also they do mention that they searched for this thing for months when this thing has been a mystery for over a decade. So,
Catopuma badia is what they're calling it.
Well, it's very exciting.
Yeah.
Very, very.
Is it Borneo now and it used to be Kalimantan?
The
or is
bay
it Kalimantan
cat,
now?
also known as the Bornean cat. Endemic to Borneo.
And also monkey cat.
Mm-hmm.
Monkey cat.
Okay.
All right, you guys. Do you know what time it is?
What time is it?
I think the monkey cat has swung us into Blair's Animal Corner.
She loves our creatures, great and small.
pet, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels. Animal Corner.
What you got, Blair?
Oh, well, you know what? I'm sorry. I have runny lips.
Ew.
Does that ever happen
What?
to you when your lips get
No.
they just get a little bit snotty?
No.
No, especially when they're super wrinkly
No.
too. My lips just right now, they're just
What?
super
No.
wrinkly and snotty.
Ew.
Does that
No,
not?
that never.
Oh, you know why? I know why. It's because I've been eating coral. Yeah.
That'll do it, I guess.
Yeah. Oh no, wait, no, I'm getting confused. It wasn't me with the mucusy lips. It's a wrasse. It's a tube lip wrasse. Yeah, so tube lip wrasses.
What are you saying?
of
Hang
fish.
on. Tube-lipped wrasse?
Correct.
Is that what this is?
It's
I
a
have
type
no
of
idea. I've
fish,
never heard of it.
wrasse, W-R-A-S-S-E type of fish. The tube lip wrasse looks a little bit different in the face than your other run-of-the-mill wrasses. And that's because most wrasses, they have smooth, thin lips, and they're kind of toothy. But tube lip wrasses, as the name might suggest, they pucker up their lips a little like this, and you can't really see their teeth. So, why? Why are they like that? Also, how do they eat the food that they eat? Well, it turns out that they eat coral. Coral is indeed an animal. People often forget about that. I always like to remind our listeners that coral is a type of animal and that they are teeny tiny creatures that live inside of a skeleton that they kind of secrete and build. And they are cnidarians, so they're related to jellies and anemones, and so they have stinging cells just like them. So if I was a fish and I went to eat a coral, I'd get stung. It would be unpleasant, and I'd probably bite down on that calcareous shell house that the coral has and hurt my teeth also. It just would not be fun to eat a coral. But the tube lip wrasse has figured out how to get it done. First, their lips look just like the underside of a mushroom. Delicious. So they look like the underside of a mushroom. They're very wrinkly. They have this kind of very finned kind of appearance on their lips. And then the second part is that their lips create their own mucus. Yeah. So the lips are like gills of a mushroom, but covered in slime, says David Bellwood of James Cook University in Australia. It's like having a runny nose, but having runny lips instead. The mucus, they think it facilitates suction while offering protection from coral stinging nematocyst. So they think that these guys go up to the coral. They kind of just press their lips up, their kind of really big lips up against the coral, and then they create suction. And they actually suck mucus, more mucus, so much mucus, off of the coral. Yeah. So they actually eat mostly the coral mucus, not even the coral themselves. So tube lip wrasses, they just love their mucus. This actually opens up a new way of looking at the way fishes feed. So we know mainly about biting fishes and gape and suck fishes. Those are the two kinds. So either a fish will bite you or they'll open up their mouth so fast, right, that it'll create suction and pull their food into their mouth. Those are the two types of fish feeding that we know about. These guys bring a brand new one to the table. They have the pucker up and suck, is what I am deciding to call it. Or you could call it the snotty mushroom lips, whatever you prefer. But it is a new way that we have now seen fish eating.
That's
so
pretty cool.
Yeah, and
It's always neat when observationally discover something that was unexpected. especially when the majority of those wrasses, they're not so tube-lipped. They're
Yes, exactly.
lipless almost. Yeah.
And there are about 6,000 fish species that we know of that live on reefs, but only about 128 of them eat corals. So they have to have something kind of special going on. And this gives us a good clue into perhaps what some other individuals are doing. So the researchers say their next step is to discover, quote, the magic of the mucus. So they want to figure out exactly what the mucus on their lips and the mucus that they're eating has going for it.
I think the magic of the mucus is going to be the title of my next solo piano album.
Oh, that's great. That sounds wet. Okay, so
let's move on to
speaking
other.
of a wet world,
Yes.
it's we did not talk too much this show about climate change. I don't want to trot over well-traveled paths that everyone has made this week out on the internet.
And
but
especially they're especially mostly political paths.
Right. But we know that climate change is real and it needs our attention. And so I think that it's important to continue looking at different ways that we can help set our planet up for success besides reducing carbon dioxide. Of course, reducing carbon dioxide should be our number one priority, but a multifaceted approach could be helpful. A new study looking at marine reserves might have an idea. So what might marine reserves have to do with climate change? Setting aside a safe haven for animals, that doesn't reduce carbon dioxide, that doesn't lower ocean temperatures, but it's actually a really important piece in the puzzle. So an international team of scientists, they actually evaluated 145 peer-reviewed studies on the impact of marine reserves. It's this study is being published in the Proceeds of the National Academy of Sciences this week, and they found that marine reserves, while they don't halt or offset climate change, they actually make marine ecosystems more resilient, and in some cases slow down the rate of climate change. So it actually can have a direct effect. Why? Well, healthy ocean ecosystems take up carbon. So if we have a marine reserve, ecosystems are unaffected, food webs are unaffected, kelp is flourishing, corals flourish, phytoplankton flourish. All those things take in carbon dioxide and make oxygen.
Well, it sounds similar to the idea of conserving Earth's forests.
Yes.
Right? You know, making these areas, we know trees are important for the uptake of carbon dioxide. We know that they are important for the creation of oxygen, but, you know, we talk a lot about deforestation. We don't talk a lot about the destruction of ocean ecosystems.
Absolutely. And so the researchers working on this paper actually, they tested the effect of marine reserves on protecting ocean ecosystems and people from 5 impacts of climate change: ocean acidification, rising sea levels, an increase in the severity of storms, shifts in the distribution of species, and decreased ocean productivity and availability of oxygen. And all of those things showed a positive impact from protected spaces and protected marine ecosystems. So essentially, keeping the ecosystem healthy helps reduce the impacts of climate change. But what's even more important is that animals that may be stressed, or ecosystems that may be stressed by a changing climate, can have a safe haven. Because really, what we're seeing with a lot of things, for example, with the sea star wasting disease, we found out it was a virus, and it's a virus that's been around before, but there were new added stressors to the equation. And we all know we can take a little bit of stress, but if all the things happen on one day, you're likely to sit down and have a panic attack, right?
Yeah.
So animals and ecosystems are just like that. They can take a certain level of stressors, but you add in all the stressors and there's likely to be some sort of collapse. So actually having these reserves creates a safe haven for animals to flee to recover, and to respond to stress in a healthy manner. Additionally, marine ecosystems, a lot of the protected areas are near or on coastlines. And when you protect coastlines, that's also directly related to mitigating the effects of climate change, because you are allowing a reduction in erosion. If, for example, in an estuary you have eelgrass, that reduces erosion. You're allowing space for animals to move. If you don't have if you're not using beaches, then animals and plants and the ecosystem can move up the coast. So basically just having protected spaces is a huge plus in the fight against a changing climate and keeping ecosystems healthy, which makes sense. But it's great that scientists actually took the time to test against these 5 factors and find that, yes, indeed, marine ecosystems that are protected, it's not just a benefit because of a reduction of fisheries or a reduction in pollution. It's all related, and actually climate change, there are benefits to protecting areas. And like you said, Kiki, this can be directly related to protecting forests or other ecosystems on land as well. It's all connected.
Yeah, it is all connected. I think the largest hurdle is convincing people that it is in their better interests. There are people in these coastal communities who rely on these marine ecosystems for their fishing, for their food, for their livelihoods. Sometimes they're watching them disappear. Sometimes when they're impoverished, they're going to be stressed themselves and have a hard time letting somebody take away their fishing area.
Absolutely.
Right? So there are lots of balances in this system. And like I said, just the biggest hurdle is convincing people it's in their best interest.
Right. And we know a lot already about that too, because a lot of these fisheries where people have been using those fisheries for a long time, they understand population dynamics, whether they know what it's called or the exact mechanism or not. They understand that you need predators, you need prey, you need to let animals breed. And so if you can kind of take it from that frame and talk about healthy ecosystems, healthy food webs, it's an easier step to take, which is why this is actually kind of exciting, is that if you don't have to talk about, oh, reduce your carbon output, oh, this kind of stuff, there's other things that people can do that are not directly related to that. If they can't afford solar panels, if they can't, if they're not in a position to change policy at this moment in time, supporting protected spaces is a great step in the right direction.
Absolutely.
Yeah.
It is a positive step. We can all hope that we can work together to take positive steps, such as this recommended one, as we move forward. And if, it's not in the United States, a federal government decision, maybe a local community decision can be made for this kind of action.
Definitely.
Yeah. Based on good science, though. That is the key.
Absolutely. And so if your local representative is not interested in reducing carbon output, maybe they are interested in protecting local wildlife areas.
Yeah, for maintenance of the ecosystems, to make sure everything was working well, that'll upkeep the economy, as well as potentially add tourist revenue to the economy. Who knows? Yeah, there's all sorts of angles we could take on this stuff.
And one idea that I came across that somebody's maybe even implementing now is energy turbines in water pipes.
Oh, yeah, that's great. Yeah, I love that.
Like, why wouldn't
why wouldn't?
Like, it's pressurized in one end to push water through. Why not have turbines in there generating electricity in the city water line?
My other favorite is wind turbines on top of freeways.
Yeah.
All good ideas. There are many good ideas. It's time for implementation.
Yes, let's do it. Oh, maybe we should go see some young innovators to see what they're up to.
Yeah,
And we
we
could
should find
find
out what the young people of tomorrow or the young people of today.
Maybe they'd have
Or
some
the young people
old
of
people
Philadelphia.
tomorrow.
Oh,
or the-
Philadelphia.
Or the young people of Philadelphia specifically. We're coming
The
for
young
you.
people of Philadelphia today, who are the old people of tomorrow, who will be
Yeah,
running.
we'll go-
Let's go to Philadelphia.
Great.
Let's go to Philadelphia this weekend.
Let's go
Let's
meet these
hang
people.
out
Let's
at
go
the
meet
Young
these
Innovators
people.
Fair. Meet people,
Sounds
talk with them
wonderful.
about what they want to change in this world, what science they're interested in. I think this is a good plan. Everybody, once again, Young Innovators Fair, Saturday, Sunday, June 10th and 11th. TWIS is going to be there. Hopefully broadcasting live. We hope to be there doing that, but we'll be there no matter what. It's time for us to end the show. We've come to the end of our show. And so I would like to shout out to our Patreon sponsors. This is a hold music. Thank you, Patreon sponsors. Chris Clark, Paul Disney, G. Burton Lattimore, John Ratnaswamy, Richard Onomus, Byron Lee, E.O., Kevin Parachan, Tyrone Fong, Andy Groh, Keith Corsell, Jake Jones, Charlene Henry, Brian Hedrick, John Gridley, Steve Bickel, Kevin Railsback, Gerald Sorrells, Ulysses Atkins, Dave Friedel, James Randall, Bob Calder, Mark Mazzaros, Edward Dyer, Trainer 84, Layla Marshall Clark, Larry Garcia, Randy Mazuka, Tony Steele, Gerald Onyago, Steve DeBell, Greg Guthman, Patrick Cohn, Ksenia Volkova, Daryl Harun Sarang, Alex Wilson, Jason Schneiderman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Robert Porter, Rodney, David Wiley, Robert Aston, Brendan Minish, Dale Bryant, Aurora Lee, Todd Northcutt, Arlene Moss, Bill Kersey, Ben Ruthig, Darwin Hannon, Rudy Garcia, Felix Alvarez, Brian Hone, Orly Radio, Brian Condren, Mark Nathan Greco, Hexator, Mish Neves Flying Out, John Crocker, Christopher Dreyer, Artyom Shishwada, Dave Wilkinson, Steve Mashinsky, Rick Ramos, Gary Swinsberg, Phil Nadeau, Braxton Howard, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Thompson, Kurt Larson, Stefan Insama, Honey Moss, Mountain Sloth, Jim DePoe, John Maloney, Jason Olds, James, Paul West, Alec Doty, Illumalama, Joe Wheeler, Dougal Campbell, Craig Porter, Adam Michigan, Aaron Luthen, Marjorie, David Similarly, Tyler Harrison, Colombo Ahmed. Thank you for all of your support on Patreon. And for anyone out there who is interested in supporting us through Patreon, you can go to twist.org and click on that Patreon link, or go directly to patreon.com/thisweekinscience. Remember that an easy way to help us out also is to tell people about TWIS. Right now, share us with people you know. Tell the person next to you that you're listening to an awesome podcast called TWIS and that they should check it out because there's some cool science in there. Share it on social media. Get the word out. Tell people that we'll be in Philadelphia if you know people in the Philly area. And on next week's show, we will be back once again broadcasting live online at 8 p.m. Pacific time. You can watch and join our chat room. Hey, chat room, you were awesome once again. Don't worry if you can't make it. You can always find past episodes at twist.org/YouTube or just twist.org.
Thank you for enjoying the show. TWIS is, of course, also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile type device, you can look for TWIS, TWIS, the number 4, Droid app in the Android marketplace, or simply This Week in Science and anything Apple marketplacey and something called Stitcher, which I still don't know if that's a real thing.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org, where you can also make comments and start conversations with the hosts, as well as other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in your subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
It's all in your head.
science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
All right, we're now in the after show. I don't know what's going. I tested my computer before the show started, and I got it all working, and then the show started, and trouble playing the music again. I do think I have a swelling battery finally. It's an old MacBook.
Mm-hmm.
I think it's a swelling battery that is
How old is yours?
5 million years. It's older than mankind itself.
What vintage is it?
Oh, can I show off something?
Big
real
old MacBook.
quick? I just got a new mouse pad.
Mm-hmm. Yay! How's it smell?
I think I have a bag for my.
Weird, right?
Huh?
It smells weird, right?
No, I don't know. I don't have a sense of smell. Hey, can we go off? I gotta talk shop. I gotta talk shopping quick. I got almost 3 hours left in my whole packing, planning, plotting, and getting ready to go thing.
Are you working tomorrow before leaving?
No, I've got children's first thing in the morning, and I'm getting to the airport because of my ride extremely early.
Oh boy.
Yeah, I have to work tomorrow, so I have to pack before work.
You guys are in packing mode. We're all in. Yeah, I'm in packing mode. I have time tomorrow to do it. And Justin, if you want to call me tomorrow, also the Portland airport, you can hang out there after landing, or you can come to my house and hang out here before we leave, and we'll go back to the airport together. They have a movie
That'd be
theater
cool.
at the airport now, I think. But
That's pretty cool.
yeah, you should come to my house. You've got
Come
hours.
pick me up.
No fair. I want to hang out at Kiki's house.
Should have coordinated
No.
your flighting.
I get a direct flight. I'm not going to complain too much. I have a question that we can discuss on air. Are we renting a car, taking a taxi from the airport, or?
I was thinking of just doing Lyft or something.
Great.
Lyft,
Excellent.
Uber. Yeah.
Okay.
Probably not taxi.
Yeah.
Yeah. But that's the
That's
direction, because the hotel is about an hour. Not an hour. It's about a mile from the event center. So we can either Lyft an Uber to the event center, or we can go for a little walk.
Yeah, I was just looking
15,
it
20
up.
minutes to walk there.
Yeah.
But
But it's let's see. Let me look.
Kiki, I need you to go. I need you to go to the flight thing from Alaska Air and confirm a seat
I am right now.
from PDX to PHL.
What do you mean?
I need you to pick a seat. I picked one.
What do you mean? I gave you a seat.
Oh, that was assigned because I didn't have the window, so I thought that was weird.
No, I don't think there was a window available.
There is now, and I just grabbed it. So
Oh,
you need to get
awesome.
next to me.
What seat are you? I was just checking
I'm
in.
10F. So if you go 10E, it's still available.
10F. Oh man, I'm like 33. Heck.
Yeah, so was I. Oh, I was right next to you. But I didn't have the window, so I changed. Did you have the window? Is that? I don't know.
I don't think so.
Well, I did. So
I might have had the window because it's nighttime, and I was gonna sleep.
Oh, is it? Oh, so
Yeah,
what's the
'cause-
point?
Yeah, because it's a red eye, and I was gonna sleep. That's why I took the window.
Oh,
But if you want the window for sleeping, you can be all the way up there.
Well, I
away
can't
from
take
me.
the old seat back. Somebody else took it.
Well, you guys are just out of luck.
You're all the way next to the restroom. You don't want that seat. It always smells like. Well, you always
I
get
do.
whiffs.
I'll be by the window. I'll be fine.
There's
I can't
whiffs.
change it because
No.
I already clicked the button.
No. I'm telling you, you can go and change right now. I clicked on their thing, and then it said pick seats, and then like here it is. I could change again if I wanted to, but right now I'm locked in.
No, I can't change it. I've already.
Well, go look at it.
No, I can't. It won't let me change it. I've already gone past that point.
What?
It didn't even offer to let me change it.
Oh, mine says request or change seats,
No,
and I've been changed.
no.
We're not sitting together now. This is
Darn
terrible.
it,
Come on.
why?
That's your fault, Justin.
Why'd you change your seat?
Well, because I wanted the window. I always need a window. It's nice to have a window.
For nighttime?
I'll even
It'll
give
be
you a window.
dark.
Just come sit next. But if you get up to go to the bathroom, I will steal your seat.
It's true. He did it.
He's like, I'm taking your seat now.
Yeah.
That's right. And then I'll be like, then
You've
I'm
got to be
sleeping.
able to do it.
on
I
you.
can still change. They'll still offer me all sorts of. I did it by my phone. I went to my phone. I clicked on the email they sent me. And then it led me to being able to do this.
Where?
Clicked on the flight.
I know. I already did my check-in though.
All right.
No, I had already gone through. I'd gotten through to all the boarding pass stuff.
Oh, I didn't do all that. Did you print something?
No,
I still
but I
have to go
emailed
stand
it
in
to
line.
myself.
I got to still go stand in line.
Why?
I'm
You
sure
can
they'll-
email it to your phone.
Yeah, use the QR code.
You can use a QR code and email
What
it to
the
a
heck is that?
smartphone.
I don't even know what that means.
It's the box. It's the scrambly box.
Or in the box.
I just have to wait 45 minutes before I can check into my flight.
Fada, I don't know. What do you mean each of the young innovators shows?
We're not going to be sitting together now if you
I
don't
have
switch.
no idea.
You should.
I sent you
really
an
switch.
email, Fada, with as much as I know. I sent you an email with all the stuff I know at this point in time. If something is broadcast through the YouTube channel, which it's very likely, it will go to twisorg.live automatically. That will just happen because that's part of the live channel thing. I don't know if we're going to use Periscope. That'll probably happen if nothing else works. And if
Hey, quick question. If I just do a screenshot of it
I don't know.
as a picture of the squiggly line thingy.
If you do a screenshot, it should work.
Okay.
But you should try to email it to yourself if at all possible.
I'll do that too.
Just in case sometimes they like want it to be bigger or something. But the screenshot should work fine.
Okay.
Yeah.
Okay, what are our plans
Party in
for
Portland. That's right, Ben.
Friday?
Friday.
You gotta come sit next to me.
So
You gotta
the
sit next to
plan-
me, Kiki.
I don't know.
Gotta
I can
fix
come
it.
sit next to you.
Change your seat.
I don't want to sit in the middle seat.
Well, that's okay. Then maybe then it was meant to be.
I've got a nice window seat that I will sleep against
Me too. And I'm
for
gonna
the
be
night.
on and off the plane before you because I'm way up front.
I am going to be on ambient.
Okay.
Oh.
Dear Lord.
Yeah, so maybe you can go because I think I might have to check bags. Should I bring up? Oh my gosh, you guys. You haven't seen this image, but I just have to send you. I don't know if it is synced to my account. I sent it to Twitter, but okay, that hasn't gone there. Maybe I'll find my Twitter tweet account. There it is. Okay, here's the picture. Oh, come on. So I have to figure out how to get this backdrop that we're going to be using for the stage to Philadelphia.
Right.
It's actually rather large.
Yeah.
Yeah. It's like
Bulky, I bet.
a bit bulky, and it was mailed to me rolled up with like around a cardboard tube and then wrapped in some stuff and taped, and I could just wrap it up and tape it again and check it as an odd size piece of baggage probably.
Uh-huh.
That's one idea. Or I can try folding it up and then it'll be
in a suitcase.
and putting it in my suitcase. It'll be all wrinkled, it'll have lines in it then, but do we care?
Well, you can air it out on Friday the second we get to our hotel.
Yeah.
And it'll be out for probably like at least 12 hours that way. So that'll be fine.
Yeah. Okay. Yeah.
that's
Okay.
fine.
Maybe. Yeah. Okay. I'm just trying to figure out how to get it. Iron. It's canvas. It's like all plastic. Yeah. So I'm trying to. My suitcase, my suitcase is going to contain this. It's going to contain the table runner.
Uh-huh.
I'm going to bring the Mevo, an extra iPad to run the Mevo. I'm going to bring also the MiFi so that we can have a spare internet connection. What else am I bringing? Oh, audio recorder for recording stuff just in case. And then I have to fit my clothes in there somewhere.
Right.
This is all going to be in a carry-on and my backpack. I'm going to make this happen. I can do this.
Yeah,
and my laptop. I have to bring my laptop. Yeah. And a couple of cell phones. I'm going to bring an extra cell phone also for just in case recording purposes.
Mm-hmm.
Recording live streaming. We'll see. It's going to be fun.
Yeah, so what are we doing on Friday?
So Friday, the schedule is going to be, we arrive in Philadelphia. Hopefully we'll get to the hotel and they will let us go into the hotel and take a nap for a couple of hours.
That'd be great.
Yeah, hopefully they'll let us come in a little early as opposed to, 3 o'clock normal time. I'm going to give them a call tomorrow and find out. And then from noon, I think it's Friday, from noon until, what time was it? Young innovators. La la la. La la la. That's not where they say. Young innovators fair this weekend. And we count down. Noon to 8 p.m. So anywhere between noon and 8 p.m., we can go and set up and check out how things work. So I don't know if our booth will already be set up for us. It should be, I guess. Yeah. So I'm thinking in the afternoon on Friday, after we've napped and lunched and relaxed, we'll go in and check it all out, and then be out of there before 8 o'clock. Get some dinner, have an adult beverage, go to sleep, get up in the morning, get back to the fair by 9 a.m. I'll actually probably want to get there by no later than 8:45 a.m. on Saturday.
Which is 5:45 San Francisco or Portland time.
Yeah, we're so messed up.
It's going to be crazy.
I envision a lot of coffee and Red Bull in my future.
Yes. I hope they have an espresso machine near our stage.
I know. Yeah. Maybe Friday also. Friday also will be gathering things like Red Bull and
double shots and
caffeinated beverages
and provisions.
A bag of snacks.
Yes. Trail
mixes, exactly.
Pretzels is.
Ooh, Snyder, you're good. A towel between the iron and the banner. That's right. We could do that. Oh yeah, and if Justin packs light, I could put things in his suitcase.
There you go. He's probably just bringing his police box.
I know.
I bought some luggage. I'm gonna be properly with wheels and the whole thing.
Wow.
Meanwhile, I usually have luggage, but I'm gonna try to not use luggage because I'm running around Philadelphia on Monday, and having luggage with me is gonna be a bit of a pain.
Eric, thank you so much for that. Yeah, I hope the Alaska flight attendants leave us alone. It's either going to be Benadryl and whiskey.
Oh,
good night, Kiki, or an Ambien. Good night, Kiki.
I'm probably just going to take Tylenol PM.
Yeah, I just need to make sure I actually fall
I did not
asleep.
think of this in advance.
Actually going to sleep so that I can wake up when the plane lands and say, "Oh, aren't we still in? Is this a wonderful new time zone? I'm so happy." Sleeping Beauty?
That was Snow White, but I think that even if you slept perfectly for the entire red-eye flight, you would not sound like that. Because you're only in the air for about 6 hours, and you're not gonna sleep perfectly during that time, and it's gonna take you a minute to go to sleep. So best case, you're getting like 5 hours sleep.
Yeah. Best case, 5 hours sleep. Oh dear.
Gonna be crazy.
Yeah. Try to go to bed early on Friday night also. Napping and going to bed early.
So, one thing we haven't talked about, which we might want to do on fast.
Yeah, we can talk about things off air. Okay, you guys.
What are we doing?
What are we doing? We got plans. We got to
Things
good.
are happening.
Things are
As long as there's an
happening.
itinerary that you're comfortable with me just walking into blindly and being able to pull off, I'm totally good with it.
We have time to talk about all this stuff on Friday. I put a sheet in our rundown
Oh.
that you can put stories in and ideas for segments into
Okay.
ahead of time, and then we can take this on Friday over an adult beverage and reorganize it and schedule it into segments. Like I'm thinking if we break up, we'll try and there are going to be a lot of amazing people at this event. And I think if we have at least one of us running around and grabbing people and bringing them up on stage for interviews and question asking, that'll be one aspect of it. And then we'll have, I think, areas of conversation. We can use stories as conversation starters, but things like the story that you talked about tonight, Justin, on the fossils found in Morocco. It would be neat to talk about, 300,000 years ago, what other hominids were walking around the planet? What else?
Totally.
Kind of go through the human history conversation. We could talk a lot about kind of the whole out of Africa thing and what evidence has maybe popped up to make us think certain things. I was thinking, because we're going to be lacking a lot of it, I've found a couple of stories about sleep, related to sleep, that would be fun. So we could talk about the science of sleep and kind of bring in stories, and anything related to space. We'll just have a bunch of stories and links that we can kind of, the new space. We can talk about SpaceX and Virgin and all that kind of stuff. We can, I don't know, split it up and, have like an astronomy hour, have a human physiology hour, have an animal behavior hour, have, mix it up. Mix it up. And take some breaks every once in a while and grab people from the fair.
So,
and that'll help fill it up.
Are we going to have a monitor to share things?
Yes, we will.
Okay.
So we can have a laptop hooked up to a monitor to share.
So there is a thing that was shared out to me that is a, gosh, I can figure out what it's called, but it's a video contest for young scientists. And they're still taking public votes for it. And that might be a cool thing to screen.
Absolutely.
I mean, it seems appropriate.
Yeah.
Oh, it's for the Bay Area. It's a local one, but it's a climate change and climate science video contest for teens.
That sounds awesome.
So, and the voting is through like the 12th or the 13th. So, actually people could still vote if they watched it at our thing.
That's a great idea.
So we could certainly do that. Yeah.
I like it. That's good. Yeah. Yeah. So the schedule, I think, is, something that we will specifically, we can talk about Ed, great new NASA astronaut class. Yeah.
There were some really interesting stories that came out this week that I just plopped on social media, so I'll have access to them that way. But there were a few stories about hearing in different animal species and new discoveries in that this week. And then there was also a really interesting story about light pollution, and we talk about light and noise pollution all the time. So
yeah, I think, common themes to things that we talk about on the show, we can bring them in. And if we ever just get in a rut, we'll just like pull out a story and talk about it.
Yeah.
And I may just be constantly pop quizzing the audience.
Yeah, and we can do audience pop quizzes every once in a while. And then I was thinking it'd be fun also to do what has science done for me lately. quizzes, just go around and get people to come up with things that science has done for them.
That's great.
Yeah.
That's a nice one.
We could talk a little bit about how to locate a credible source.
We absolutely.
I get on my teenage volunteers about that all the time, talking about being a skeptic and how to figure out where sources get their information. So we could definitely do a quick crash course in that. That might be fun.
Absolutely, and we can maybe bring up part of the Skeptic's Toolbox or some of those websites that talk about arguments of logical fallacy. How to recognize logical fallacies in arguments.
I love it.
Yeah, that's all part of that. Yeah. I mean, I can even take some time, I'll sit down and talk about bird brains.
And if we're interested,
research I did, we can talk about food science. We can talk about,
we have options. We can talk about it on Friday for sure.
Yeah. What I really want to do is like Friday while we're there, like getting the booth set up, making sure everything's done. Then I want to see who's around and maybe like grab people and be like, hey, you want to come by for an interview tomorrow?
Mm-hmm.
Kind of nudge people, find out who's around.
Yeah, that sounds great.
Yeah. I think we can fill up.
We can do
8 hours of podcasting.
We did TWIS-mageddon.
So no problem.
So, that's really nice having that under our belts because it does make it feel like it'll be fine.
Everything is easier than a 21-hour show, was it?
Yeah,
and
21 hours.
Here's an option. If the internet is holding up and we can do a hangout from where we are, I don't know how it would work exactly if we could make it work, but if we could do a hangout from where we are, we could even invite people to join us.
Mm-hmm. Yeah, we just have to have a speaker system.
Yeah, that's what I'm worried about. I'm not really sure. They're like, you don't have to worry about it. We'll have everything for you. All you have to bring are your laptops. I'm like, really? Okay.
I'm just bringing my power cord.
Yeah.
I
definitely bring your power cord.
But I don't need anything else, right? I don't need any other hookups or
bring your laptop.
Well, that's what the power cord is for. Okay. And so we're gonna wear our TWIS shirts, so I don't need to worry about packing special clothes for that.
Yeah, no special clothes, just TWIS shirts. And...
Okay. Yes, please try to bring me a t-shirt.
Okay. I will do that.
Okay. And
yeah, if we can see twis.org, the chat room could ask questions. Yeah, I mean, I can always open up the chat room, but it all depends on whether or not the internet situation allows us to live stream, which is what I'm really concerned about. But we're a podcast. So regardless, I can record it, which is good.
Mm-hmm. Mm-hmm. And at any point, if you just want me to vamp about anything particular relating to animals, I'll be good to go.
Exactly. No, we're fantastic there.
Blair, why don't you tell us about different types of mammal skulls?
Well, we have hours later action here. Hey, Blair, do you think animals are intelligent?
No.
Give me examples of animals not being intelligent then. What are some of the unintelligent things that they do?
Right. Yeah. So yeah, it'll be fine.
Yeah.
It will. Hey, Snyder, I don't know if I've had Fraser. I have done work with Fraser before. I think we've had him on the show once, a long time ago, I think. But we don't, and I've had Pamela Gay on the show once, but we don't work with them regularly. We could do more collaboration. I do know Fraser and Pamela though, so that would be fun. Punderful puns. Oh, punderful. Right now, I'm a video podcast. That's right, Hot Rod. Okay. We can talk about astronauts.
Okay.
Right. There's some, I mean, seriously, stick us in chairs next to each other for 8 hours to talk about science.
Yay!
It's gonna be amazing.
I know. Ask me if the Great Barrier Reef is dead. Then I'll probably start yelling.
Yeah. Oh, and we can talk about the science of climate change, and we can talk about ocean acidification, and we can talk about, how we know ocean acidification is taking place and what the problems are for species living in the ocean. And then, even the stories that we talked about on the show tonight, we can bring your marine eco preserve story and how does that tie in with carbon sequestration. And that's like a half-hour conversation. Easily.
I mean, I do a 90-minute presentation for volunteers at the zoo on how to talk about climate change. So,
and that's just how
to talk about it.
Yeah.
So there's lots of opportunities.
And we can talk to Justin about the psychology of selling cars.
There's none involved.
That's not true. The glasses.
No psychology involved.
Okay. Now,
nothing to see here. Move along, everybody.
Recognizing I'm probably going to wake up at 5 tomorrow and then red eye the day after that.
I gotta go. I'm still gotta pack everything now, then go pick up a child as soon as I wake, and then I'm going to be almost directly to the airport.
Yeah,
so.
And I want you guys to know I found the monkey cat before anybody else did.
Yes, that's a monkey cat.
There's a monkey cat.
I just looked a bunch trying to find information about the bay cat, and apparently they knew that the bay cat existed in like the 1800s.
Yeah.
And they know the exact genus and species, and they actually know the exact date at which it diverged from the common cat lineage, which is very interesting. So
I went this one. Where'd that one go?
I really
banana.
wish though that it was called the bay cat, like BAE cat. Bay, you're my bay cat.
This monkey cat looks so sad.
Oh no, monkey cat, no like bananas.
Sad monkey cat. Okay, enough of the monkey cat. Okay, you guys, let's go. You guys sleep, pack, do the things you need to do. I will see Justin. We'll see you tomorrow. Give me a text when you are leaving and estimated landing time or flight number so I can track it. I think I might have it in here somewhere, but yeah, that would be good because then I can maybe pick you up at the airport, and then we'll take we'll take a car back. Yeah, that'll work great. And then Blair, we will track your. I have your flight information because you sent that to me, so I will track your flight, and if you're landing approximately same time, that hour window, Justin and I will hang out, get a coffee, have breakfast type stuff if our stomachs aren't too upset. And then we'll hang out for an hour or so and we'll see you at the airport. If not, we'll see you at the hotel, but I will text you if we're leaving.
Yeah. We have telephones.
We have telephones to coordinate, but that's the plan as far as it's going right now. It's actually happening. Oh, good gracious.
I'm really excited. I think it's going to be great.
I have a very large TWIS banner.
Hooray! That's all you need.
Oh, I need it so
Step
great.
1, branding. Step 2, profit.
Step 2, do something.
Step 3, profit.
Step 3, profit. There we go. Yeah. All right, you guys. Thanks, Snyder. Thanks for everyone watching tonight. I hope we do have a safe trip. We all want to get there safely, right? And have fun in the process. And hopefully we'll talk to a lot of young innovators, young budding, inspired scientists, and we'll be inspired by them. That's what I'm looking forward to. Yeah, Adrienne, Godschalx. We'll link to it on twis.org if you want to, if you missed all the links earlier. she was a great guest. She's wonderful. And everyone in the chat room voted that she should be a guest host at some point in the future. I thought she was great too. All right. Good night, everybody. Hey, say good night, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good night, Kiki.
Good night, Kiki.
Good night, Kiki.